Yeah, that's a great question, Celeste, and thanks so much for asking it. I'm going to take a step back and give a little bit of background, and then I'll get to your question. So, a little bit of a further introduction. Like I said, I'm Anushua Sinha, and I'm a Med-Peds trained infectious disease physician. I'm the lead clinical director for the Influenza program, which is a real honor because in my previous life, when I practiced pediatrics, needless to say, there was a lot of RSV in the office. So, as your audience is really well aware, RSV is the number one reason why infants are hospitalized everywhere. And we're in a position with influenza to help reduce that burden of RSV in infants. Influenza is Merck's preventative, long-acting monoclonal antibody for the prevention of RSV in infants born during or entering their first RSV season. And the FDA approval for influenza was in June last year, June 2025, followed by AAP and ACIP recommendations for its use. And that's been the culmination of over a decade of extensive research here at Merck Research Labs. So we're super proud of it. Influenza has a strong clinical efficacy profile, which shows significant reductions in RSV disease incidence and hospitalizations, and it has a strong safety profile as well. And we published the CLEVER and SMART trial results last year. I think what you're talking about here with your question are the final SMART season 2 results that we just presented at a global RSV conference last month. So thanks for your question. So let's talk about the SMART trial. As I just mentioned, SMART was one of our two pivotal clinical trials, CLEVER and SMART, that supported that FDA approval of influenza that I just mentioned. And as doctors experience everywhere, all infants are at risk for RSV, regardless of when they're born, how much they weigh, or whether they have underlying risk factors. SMART is our trial that focuses on that group of infants with those underlying risk factors that render them at increased risk for severe RSV disease, right? Those infants who are early or moderately preterm, those with chronic lung disease, or with congenital heart disease. So that's the focus of SMART. And in SMART, all the children who received influenza in their second RSV season were at increased risk for severe RSV disease as they entered that season, which means that nearly all of them, about 99%, had either chronic lung disease or congenital heart disease. So that's the SMART trial. Focusing in on your question, when pediatricians are thinking about the protection that influenza offers young children who are entering their second RSV season with a continued need for protection because of the risk factors we've been describing, they'll need to turn to the results of the SMART trial. And it's important that they first consider how influenza works, right? Influenza is a monoclonal antibody that targets a viral protein, RSV's F protein. It doesn't interact with the host by design, which means that if you achieve the same concentration of influenza, then you can bridge the efficacy that was demonstrated in CLEVER, our pivotal phase 2b/3 safety and efficacy study in healthy infants. And in fact, that's exactly what we saw in SMART's results for season 2. Those efficacious concentrations required to be achieved in the second RSV season were achieved following administration of influenza. Yeah, you're getting right to the crux of the matter with your question. Let me just say another word about the methods in the SMART study. So in SMART, in the first RSV season, infants were randomized 1-to-1 to receive either a 105 milligram dose of influenza or monthly palivizumab. In the second RSV season, those eligible participants that we've been discussing, children under 2 years of age with chronic lung disease, congenital heart disease, preterm birth, or certain other risk conditions, received an additional open label 210 milligram dose of influenza. So the second season dose was 210 milligrams. Now, as you're pointing out, it's important to think of monoclonal antibody serum concentrations or exposures to influenza through the full 5 months of a typical RSV season, which means you need to think about these exposures as concentration versus time. And in fact, that's what I'm referring to when I talk about efficacious concentrations. To get a little bit technical, what I'm talking about is the comparability of the area under the curve from days 1 to 150. And that's the key metric that we're talking about when we're talking about bridging using concentration.